Okay, hi everybody. My name is Ethan Moyer, and this is April 8th, 2021. This is our second Linux workshop I'm holding for Zhou Lab. And today we're going to dive deeper into more awk commands, as well as a little bit of tbmate, and then I have one bedtools example that I want to get through. So I want to start off with some things I left out when I was talking about AWK. So I have a prepared directory that I'm just going to work in. There are some files in here, and that should not be one. Okay, so we have two files. And what I'm going to do is I'm going to start with some data. So you can use tbmate view, and you give it the tbk file, and you can view the data in that tbk file. And this is formatted as a bed file. So what you can do is you can redirect its output to bed file. And when you look in the local directory, you'll see that the bed file is right there. A bed file because it has genomic coordinate. Well, this is the same format of a bed file, but in all actuality, when you have a bed file, you want the first three columns to be the chromosome locations. But when you're using an array, you typically have the CpG probe identifier. And then these middle two columns are placeholders, and then the fourth column is the beta value. And then with bed files, there's a whole slur of, if you look up bed file format, there's a whole slur of column IDs.
Can I have a question?
Yeah, sure.
I don't think that's a beta value. The beta value cannot be negative, right?
Yeah, sorry. So -1 implies missing values. There is a, yeah, sorry about that. There is a setting in here. If you do tbmate view, you can change it from -1s to NAs, and I think that's -N, but I don't know. I'll try that out real quick. So that would be go before, after the view, but before the file name. That's obviously wasn't it. Use, it should be like use provide. There we go, -D. Sorry. So whenever you have negative values, that means that there are no beta values for that probe location. So you can just have those as NA. Good question though, because that brings up a setting for tbmate. tbmate. And so really when you look at the bed file format, this is just on Wikipedia, you can see that we have different columns and what they correspond to in the definitions. I'll actually put this here, info on bed file. But when we throw around the name bed file, it's okay for it to look as simple as this, where the fourth column is reserved for data, and then the first three columns are reserved for either probe assay identifiers or genomic locations. Okay. So I just want to review a little bit about AWK before I start throwing around some stuff. What we can do is we can use AWK, and when we do AWK, it tells us all the options, which I'll be going through in a bit. Not all of them, just some of them. And then the structure of the script has a beginning, body, end. I said this last time, the beginning can be thought as variable initialization. This is what you execute per line of the file you pass in. Make it bigger. And then end could be thought of like printing something at the end. So if you wanted to form a column average, you might have a running sum of each column, and then you'd print out the average, which is just the sum divided by the amount of entries in each column. And then you can pipe that to another program. You can also use less to read the file, pipe that through some function, read that in through AWK, and then print it out through, or pipe it to less. So when we use awk on our system, you'll notice that this also has an alias that I really didn't explain. There's a couple of variables here, and we can see all of these variables either using help and awk dash help. And I think the short command would be just -8 -h like that. Or you can use man, which stands for manual, and this is just like less, where you can use Z and W to page down and page up, and then you can see the options that way. So when we do alias AWK, you'll notice that there's a -f command, a -f parameter, and that specifies the delimiter of the file, right? So we can say, just for kicks, that we don't like TSV files, we like PSV files, pipe separated value files, and those aren't real. But we can use sed to form a replacement that'll replace all the tabs in that with pipes, right? You can see that instead of there being 3 tabs, there are just 3 pipes on each line. We can use sed to do this by doing a global replacement, and the sed command is usually, you have sed, and then you have 3 forward slashes. What goes in between the 1 and 2 is what you want to replace, and what goes in between 2 and 3 is what you want to replace it with. And then after the last forward slash is a toggle for whether you want to do the first match, you want to replace the first match, or you want to do a global replacement. So that's how I generated that. And then you can just ship this off to a PSV file, which I have, which I totally just made up. And if we were to read this in using AWK, it wouldn't recognize that the columns are separated by a pipe, which is just the vertical line. Instead, we have to change the delimiter to the pipe, right? And then we can use AWK to print out the first column. So if we do this, you'll see that we print out all of the probe identifiers for the assay, for, I think this is epic, right? And there's also something in the alias. You'll notice that there's a V and a OFS. Basically, the V means that you're specifying a variable, and then OFS is it stands for operator field separator, I think. I'm going to be talking about it more later. Output field separator, yeah, sorry. So let's start off with the idea of just giving it a variable. So you can specify a variable from either like a string or bash. Here I'm just giving it a string. And we can say that that variable space E is equal to this probe colon. And keep in mind that we're still dealing with a PSV file. And we want to print out that variable, which we stored in E, and then the first column. And right here, I'm using printf instead of print. Printf allows you to specify, the first parameter is a string where you can have %s to specify string input. So each one of these string identifiers corresponds to the variable. So this corresponds to the second argument, this corresponds to the third argument. \n is a new line, and the space here is recognized as a space. So if I were to put this through less, you'll see that we have the variable that we stored in E printed next to each of the, probe IDs, right? And we can go a step further. We can assign this in bash. We can say E is equal to probe, and then we can even do it this way, where you have a command wrapped in parentheses with a preceding $sign. and you can store the results of this command into E. So you can do echo $E to see the contents of E, or you can do well, on the other hand, when you do $E, sorry, when you do echo E, this is wrapping and I don't like it, sorry. There we go. When you do echo E, you'll notice the difference between echo $E and echo E is that when you include a $, you get the contents of E, but when you do echo E, it just prints it like it's a string. So you can kind of think of the $, you're telling bash to grab the contents of the variable E. So it's kind of like referencing that variable. Okay.
All right, I have a question.
Okay.
So awk dash capital F, and you did the pike, is what's like the default setting for reading the files? Would it be able to like recognize a TSV file, or would you have to specify \t if you're?
So that's a good question. The reason why Wanding has this for it's -F \t because the default setting is a space, and you can see that here. There's dash capital F or something, field separator. It might say it here.
So you're saying by default it is \t.
Yeah.
So it is. That's why we have a lot of TSVs.
Yeah.
Okay, that explains a little bit there. Cool.
User. Yeah. What I was trying to do is I was trying to see if I could find out if that's actually right, but I'm almost positive that the default is \t. The default is space, and that's why we use the -t's. Okay. So when we look back at the alias of AWK, that's set. This is set in Wanding's bash RC, and it was included in the lab HPC tutorial to kind of copy that into your local HPC, or at least source it from your bash RC file. And I can go into details about what specifically that is and what that means later. But by setting that in your bash RC, which is just a file that sets up commands that want to be executed on login, it makes this an alias. And then the other thing is that he's not only setting a variable, but he's setting a variable for output field separator. So this is what is output, and this is what awk uses to separate variables. So we're not using printf here anymore. We're using print. And we included a comma between the two. So when you include strings or variables to the print statement with separated by a comma, it'll basically fill in whatever it has for its output field separator. It'll put it in between these variables. So the default with our version of AWK is \t for a tab. So when we do this, you'll see that all of them are separated by tabs. But if we were to say, okay, we want to have -v, and we want to say that the output field separator is a dash, you'll see that this quickly changes so that we have dashes in between E, which is a variable we stored, which is just probe, and the first column, which would just be the probe ID. And playing around with just this one line tells you a lot about AWK and how it works. You can give it variables. You can give it variables that have specific interpretation to AWK. You can change how you recognize the file. You can recognize the delimiter. It changes how you recognize the delimiter. If I wanted to change this to the TSV we have stored, or really it's really a bed file that we have stored in this directory. All right, because we have this bed file that's really a TSV file, and I want to recognize that TSV or bed file. I would have to change this to \t to do that. but we're using PSVs. Yeah, okay. And I was showing how you can even do something funny. You can say, okay, right, we're storing probe in E, that's the string probe, and we're recognizing PSV files, or we're recognizing files with delimiter as a pipe. And then we want our output field separator to be a pipe, right? So all we're doing is we're putting a pipe in between the string stored in E, so probe, and the first column. So you'll see the output is like this. It's not that you'd ever use a PSV file, it's just to show you that you can change variables that has meaning to awk, but you can also use variables that you might store in bash. Does that make sense? Hopefully. If you have any questions, just call out. All right, don't bite. You can even change where you initialize the output field separator by using the beginning block. Remember I said that AWK can be formatted such that you have beginning, and this is for variable initialization. Instead, what you can do is you can say, okay, let's initialize the output field separator to be a pipe in the beginning block. So this will execute before you even start reading the file, and then the body block will execute for each line of the file. So we'll see that this generates the exact same thing like this. Okay.
I have a question actually.
Mm-hmm.
So if you wanted to set multiple variables in the very beginning there, would it just be like a comma, or would you have to do
Semicolon.
semicolon? Okay.
That's a good question. I didn't mention that. See, some languages you don't need semicolons. Other languages you do. In general, it's one of two cases. I know in R you don't, C you do, Java you do, Python you don't. But with AWK, it just so happens that you do need them to separate them. You don't need to put a comma after, you don't need to put a semicolon after the curly braces, just after like variable initializations or print statements. So if I were to do a print statement and then follow this with a for loop of some kind, then I would need a semicolon, but we omit them because it recognizes that it's the end of that block. Okay. Something useful that actually just recently came to my attention, Wanting told me, is that we can actually create functions for AWK. So let's say that I wanted a function that only gets odd rows of a PSV file. Okay. So we pipe in a PSV file right here using less. We send it to the function, and then we print out the result. We're defining this function as such. You have the name of the function, these open brackets, which might either be just for syntax to identify that it's a function, or for arguments, I'm not sure. It's one of the two there. And then you have open and close curly braces. In between, you have just the awk command followed by a semicolon. So what we're saying is we want it to recognize PSVs, pipe separated value files. And then we're saying if the row number, NR is the current row number it's on, is not divisible by 2, or when you divide it by 2, you get a 1, or it's an odd, or basically NR is odd, is what this is saying. For anyone who doesn't know, this is modular arithmetic. You're asking, when we divide the row number by 2, what's its remainder? When its remainder is equal to 1, then print out the line. The closest thing that you'll deal with in modular arithmetic in real life is time of day, right? I think in the US we use a modular system of 12. So after 12 hours it resets, and it does that twice. And we specify whether it's the first half or the second half of the day by AM or PM. So then after you define this function, you can export it by doing export -f. And I think that -f flag makes it seem like it's defining it as a function, or tells awk that it is a function, and then the function's name. And then what we can do is we can pass this file, which is a PSV, right? We can pass it through, and you'll notice that in this file, the second line here has CPG that ends in 103. And when we use this one, you'll see it skips 103 and goes to 109. Okay. So what if we want to make a function that changes a PSV to a TSV, right? So PSV to TSV, and TSV in our world is also a bed file because it's tab delimited. What we can do is it's the idea of saying, okay, let's take all of the columns for a given row and let's separate them with a tab. That's like what I'm thinking in my head. So then really what we have to put in this command here is what we have to do for a single row. So we're doing a for loop here. So we're saying for I equal to 1 because the first column is $1 to NF, which is the number of fields in a given row. We're saying for each of these from 1 to NF, print, you're going to use printf, print that column, and then append a tab, and then $I will just grab, it'll grab $1, which is the first column, $2, which is the second column, $3, which is the third column, all the way up to $NF. So here's an indication that you can use variables for accessing. If you have a variable that's an integer and you want it to use it to access a column, you can just put it behind a $, and you'll access that column number. And then when we use print by itself, it'll automatically print a new line character at the end. But when we use printf, we have to say, okay, after it's finished the for loop for that line, we want to print a new line. So the next row is printed on the new line. So we can export that function. And then we send the PSV to the PSV to TSV. We surprisingly get a TSV file, which we could rename to be a bed file. So I think that's it for what I wanted to do with AWK. I was going to go into just some things with tbmate, but do we have any questions with either the output field separator, variables, the delimiter variable? I introduced NR and NF and how they can be used. Talked about print and printf.
What does the percent usually do?
That indicates that you want to, okay, you can think of this, there's a name for it, and I don't know the name, but this basically specifies that you want to replace this with a following argument, right? So this %s, the percent sign means that you want to so in some languages you can do like %f to specify that you want to insert a float. In other language, and it might also be that it'll be %d to indicate that you want to insert a double. %s, you can kind of think of inserting a string. So right here, we're going to insert, this corresponds to the first following argument. So it corresponds to an E. So you can think of whenever you see %s as placeholders for following arguments, like E is a string, and this happens to be a string, so it's put right there.
Okay, and also for creating a function like the PSV to TSV, are there like limitations to a function you can create, or like rules you have to follow, or can you really do anything?
You can do anything, but like one implicit function is that it can only act on one line at a time, is what I think of. Okay. Actually, that's false. When you're making the body of this, you can have it act on one line at a time, but I guess with functions, if you really wanted to, you could have the beginning and you could also have the end right here. I could put them in there and I could run it. It would be fine. It wouldn't do anything. But so with this, there really isn't any limitations on that. There is no limitation. It only has to work on a line at a time. Sorry, because all you're doing is you're inserting an awk you're inserting an awk script, which is really what all of these are, into kind of like you're kind of wrapping it around a function name, and then you're exporting that function name into your bash environment, and then that's what you can ship input through. So you send in the input, And it's almost like, it's exactly like if you were to actually put the awk script here. So if you were to take this and put it there, that's kind of the same thing. It just makes it cleaner. And if you want to use, if you have a bunch of PSV files laying around, you might want to convert them all to TSVs, right? Or if you were some psycho and you really wanted colon separated values like probe, number 1, 2, you could do a TSV to colon function, but I really don't see the point in doing that. But good questions, yeah.
All right, thank you.
Yeah, just kind of hoping with these more details about AWK would give you a deeper understanding for how the function is working. And then once you know, yes.
Could you please explain what's the difference between the 2 pipes sign on line 57?
Yeah, okay. So you can think of the -f as what you're reading in, and you can think of OFS as the output field separator as what you're printing out. So for F, by specifying that -f is a pipe, that means that you want to recognize in the input, in standard in, or the file that you give it, that each column is separated by a pipe when you read it in. And that's what allows you to access each column. If I had a tab here and I had a tab, I can show you actually. If I had a tab here and I wanted to print out E, which is probe, and $1, this will actually print out probe and then the entire line, separated by a pipe. Because, as opposed to doing this, because this is a pipe separated value file, right? So we want to recognize that each column in the file is separated by a pipe. And I could change this to a tab, right? And it'll just be used when you're separating arguments, when you're separating variables when you're printing them. So -f is for reading in, and OSF is for printing out.
Okay, I see. So you just replace the pipe to a pipe, like.
Yeah.
Okay.
Or I can replace the pipe with a tab, right?
All right, I see.
Yeah.
So what's the difference with the line 61? Is that also replace the pipe to a pipe?
Yeah, they do the same thing. It's just showing you that you can either specify the output field separator, which is specific to awk. It's used by awk implicitly. Notice how I use E in here, but I don't use OFS in here. That's because you can kind of think of it like a path variable, even though it's just not that. Awk recognizes OSF and then it applies it behind the scenes. You can also specify it using -v outside, or you could specify it in the beginning statement inside. So it's just 2 variations of doing it.
Okay.
Yeah, good
Are
question.
there other variables like OFS that awk recognizes?
Yeah, there are, and I used one a while ago, and I just don't remember. I had some special use for one, and I just can't remember what it was, but they're out there.
It's in the manual, I suppose.
You want to go on a hunt? We go hunting. Maybe if I just do a. Okay, built-in variables. Here we go. All you want to know: field width, file name. You can specify the input file.
Mm-hmm.
Okay. There's an ignore case. That's the one I used. If you wanted to ignore case when you're doing, awk also has functions you can use inside of it, right? It has a match function, which might be useful for matching characters or matching words. And it also uses this utility, which is a way of doing kind of like a grep. And a grep is just matching a string to a grep. Is a Linux command that matches a string to a regular expression, and a regular expression is a way of generalizing patterns. So for instance, you might have, if this would be in the awk, you would have if the first line contains CG that you'd want to print it, right? And that's how you can use the tilde to do it. So specifying ignore case equals true at the beginning allows you to ignore the case. So this would recognize capital C, lowercase p, capital C, lowercase, capital C, capital P. Yeah, but good question. Plenty of other ones. And that's the one, ignore case, I used. NR is NF is the number of fields in a given line. NR is your is the number of records that is like the record number that you're currently on, or the number you've seen so far. There's also another one called FNR, which is the number of records that you've seen for a given file. So if you pass multiple files into awk, it has a way of telling you have a way of knowing whether you're on the first file or the second file based on whether FRN equals FNR equals NR. Because if this is true, you're on the first file. If this is false, you're not on the first file, so you're on the second or third file. So then if I were to do like awk some script, file 1, file 2, it'll go through all the lines of file 1, and then it'll go through all the lines of file 2. And what I can do is I can say if FNR equals FR, execute this line. And so this block will only occur for file 1, and then I can be specific for file 2 if I do this. Basically, when they're different, execute it for file 2.
But you wouldn't be able to extend that to like 3 files or 4.
I don't know if awk takes in 4 files. I don't know if awk can take in multiple more than 3 files. With all I've done with awk, I've never had to give it more than 2 files. Is what I'll say. You can Google.
Yeah.
That's how I learned all this.
I'm
Okay.
sure you can probably like loop or something if you have multiple files.
Yeah, you can probably do that.
Like you're doing multiple, but so like what about columns? I see like you have row numbers as the $. How would I specify columns if I could do column numbers? If that, I guess you wouldn't be doing column numbers because it's done.
Sorry. Okay.
Yeah.
NR is to control the row numbers, right?
Yes.
You specify the column numbers using the $.
Okay. Because I think you might have said something a little different last time.
What did I say?
I thought you said the $1 was for the row number 1.
Oh, yeah. No, that's for column number 1.
Okay.
Did I misspeak? I'm sorry.
Maybe you misspoke, or maybe I just like am confusing it with something else, but who knows?
Okay.
But yeah, that's good to clarify.
Yeah, sorry. Yeah, no, the $ specifies the column number.
Okay, cool.
And then NR and FNR can be used to keep track of the row number. Okay. So we're going to take a look at something that I had Mike look at a while ago. So here is a RNA-seq data sample. Basically, the first column is a gene ID. It's an ensemble. It's either an ensemble gene ID or a gen code gene ID. I think it's an ensemble gene ID.
It's ensemble ID. I've used
Ensemble ID. Okay, so it is an ensemble ID. And for each row, you can see that we have an ensemble ID, the corresponding cell type it's taking a measurement of, and then column 3 specifies replicas. So if, I guess they have, they have up to 6, it looks like they have up to 6 copies of a cell, and they're taking measurements of TPM and PTPM, which are measures of expression. They're measures of RNA. They're measures of expression from RNA-seq. And really what I think is just the one thing that matters, higher the number indicates that there's more expression of that transcript or that gene. So these correspond to gene IDs, or these correspond to genes or transcripts or RNA transcripts. Okay. So why do I bring this up? I bring this up because, right, if we're downloading data from the internet, we kind of want to have, like, there could be various ways that we can represent it, right? We have this file. This is the raw data from the internet, but we can't exactly access the data very easily. We can't say, okay, so I want to take all of the gene IDs. I want to find all of the expression data for the genes that are in this chromosome region. What would be very helpful is if we mapped this, or we created a TBK file for this data, okay, using tbmate. The problem is that we have to reformat the data, one, and we have to create an index file. An index file is what maps genomic coordinates to specific identifiers like these transcript IDs. Okay, so what we're going to do is we're going to grab all of the, oh, that was bad. We're going to grab all of the transcript IDs or gene IDs, and then we're going to sort them and send them through unique. And what this does is it removes duplicates. When you sort them, it puts them in order, A, and unique removes duplicate entries. So it'll only print the first occurrence when something is sorted. So this is going to take a long time, but you see that we have unique values, and then we can store that in a file. And what we're going to do, and I'll explain this later, is we're going to take those transcripts and use a function, use a library in R called biomart that effectively maps those maps those transcript IDs to their genomic coordinates, given some ensemble version number, because it just so happens that these ensemble gene IDs kind of change a lot depending on what version you're using. And so we can take, this is the output that we get from that R code that I'm not showing. And what it does is it takes all of these are in the same order of which we sent them in. Like we started with 3 and they were ascending. It maps that ensemble ID to its genomic coordinate, and then it records the line that this ID appears on. So we know that this was the first row or 0th position. This is the second row or first position. And then what we can do is we can sort this using sort. So we want to sort the first column lexicographically and the second column numerically. So if we look at the file again, we'll notice that the first column is chromosome numbers, and it just so happens that in convention you sort those lexicographically. So 1 comes before 10 is the idea. Sorry, that's a bad example. If I just look at it, you'll be able to see. One second, I'll show you what I mean by lexicographically. Like this, or actually I'll just use cut-f f1, type of C unique. This is what I mean by lexicographically. 1 comes before 10, but then it's 11, 12, 13, 14, 15, 16, 17, 18, 19, and then 2. That's what I mean by lexicographically. And in convention, this is how bioinformatics people chose to represent this stuff. So we do that sorting, and we use bzip. If you go to bzip-help, it doesn't tell you what it is, but it's basically a way to compress files. And we want to compress it because that's what the tabx file recognizes. So at the end of this, what we create is we create an index file that's sorted by the first column and then the second column. And then in the fourth column, we have at what line does this transcript appear on in the raw data. And then you can use I don't understand these commands, but you can use tabx to create a .tbi file. So if I look in here, if I go to this directory and I hit tab again, do that, .tbi, it basically, using that tabx command creates a TBI file, and you need both the both this file and the TBI file for tbmate to work. And I think that the TBI file is the binary file, is kind of a binary file that tbmate uses. So you encode the non-TBI file, the one that we generated, the one that looks like this. You use this, send it to tabx to create the TBI binary file, and that's what you use in tbmate. And then you can use both of these things, and the data that you created, you would take that. It's a long process, but you take this index file and you create bed files out of all of these, and you can use the index file to index the TBK files you create from the bed files that you create. So the process goes: raw data, bed files, TBK files, and then you need to create a index from the sample, from the data, that fall in line with what data is stored in the TBK files, and then you can use the index file to view the TBK file. You can ask questions. It's fine. I see. You have a face on yourself. You have a face, Elaine. What are you thinking?
Sorry. Could you explain the columns again? What the numbers are in the files you created? Like usually when I see something like that, they're like start and stop positions, but that's not.
Yeah.
Is
Yeah.
it?
So right here, you're talking about this one.
Oh well, this. What are the letters?
The letters specify experimental replicas.
Okay.
So you have a basophil, but you have 4 different types of basophil you're getting measurements for.
Can you say that again?
Yeah, sure. So you're looking at the expression of this gene in the cell type basophil, but you're doing like multiple trials. So like for instance, you'd expect that these values would be similar across different trials because it's the same type of cell, but they're just biological replicates. I don't know if biological replicates is a good way to say it. They're basically doing, for basophil, they're doing 4 trials and 4 separate basophil cells.
Oh, okay, that makes
Yeah,
sense.
it's a better, yeah.
And then in the other one, there were 3 columns in the middle. Did you say those were the start, stop, or yeah? What is that?
Yeah. So, oh shit. So right here, do you understand how we got to this point?
Yes.
Okay. We're taking this. What we do is we take this file into R, and we basically map each of these IDs to where they appear in the chromosome or in the genome. Yeah, good word. And that is what gives us this file, right? Because this is all ordered, and the first 3 columns here are their genomic coordinates.
Okay.
And then this just corresponds to the index of the line. So this is index 0, this is index 1, index 2, index 3, index 4. You see how it's just increasing?
Mm-hmm.
Yeah. And then we take this file and we sort it. The first column is prioritized to be sorted lexicographically, and then the second column is sorted numerically. So you can see that we just have chromosome 1, and then these numbers increase in the second column.
Okay.
And then these all get shuffled around, but their position in the data is still conserved because we have this column right here.
That makes sense. Thank you.
Yeah, so what you do is you go from raw data. I'll do general idea, raw data to bed files to TBK files. The thing is, what's interesting is you don't need the index file to create TBK files. You actually pack the TBK files independently of creating the index. But what you can do to create the index, you go from raw data, and then you want to grab relevant rows. That's what I generate in the samples.txt. It's just a sorted version of all of the unique IDs, and then map to genomic coordinates. I did that right. Map IDs / rows to genomic coordinates. And then you want to sort and compress. And then we want to send to TABX. And if you want to see the specifics for how, I kind of left out the whole R biomart here because it kind of delineates from the idea of doing everything in Linux right now. You can look up like this file name or like this file name in my lab journal, and it'll show you the specifics because I have all of it saved there. Do we have any other questions from anybody else about this setup? Because although you probably won't be creating TBK files for RNA-seq data, you might be creating it for other type of data. And luckily for all the data that Wanding downloaded, he kind of automated this process. But it's good to understand how tbmate works, and doing this process kind of allows you to understand it like that. And then here, this might allow you that you do tbmate view when you want to access the you want to view the TBK file. You can actually give it a optional argument right here, I, which is just path to index. So the index we created right here would go after the I, and then the TBK files you create from the bed files would go right here. And then if you want to, and how you go from bed files to TBK files, you do tbmate pack. And this is a representation of how you would do that. Path to bed, and this one would be path to TBK. And that gives you the basic skeleton for how you go about doing this process. Okay. So for this section, for tbmate, any questions?
And index files are just made with the biomart in R.
Yeah, the biomart makes these index files, and then you use TABX. I don't understand the process of TABX as well as the other steps, but that's just something Wanding A told me to do, and B, it creates a binary file that's kind of like a companion. You can think of it as like a companion file with the text version, which is just this zip file right here. Oh, something else I want to say, when you have .gz files, that means it's typically zipped using like bgzip or gunzip, which are just different algorithms. And if you wanted to look at the line numbers of the amount of files, what you could do is you could do this, and this will tell you how many rows there are in this index file. So this is useful for getting like the number of ensemble IDs that any TBK file will be mapping to. But if you do this, if you do this, you see how this number is so much different than that number, but we're effectively looking at the line count of the same two files. It's because that when you do LC and this is getting the number of rows of binary. And in order to unwrap the binary, you have to put it through LS first. sorry, you have to put it through less first and then send it to LC, which is a line count. And if you're curious, the alias of LC is just WC, which stands for word count, and -L, which gives you the number of lines. That's another thing. Tons of aliases, but I'm getting used to them. I'm actually getting used to the aliases before I get to used to the regular code. So, okay. So we have some time left, and I just want to talk through these couple examples about bedtools. So bedtools is powerful. It works on bed formatted data. So data that's formatted like, that's a bad example. Data that's formatted like this. Okay. But not formatted like this. Formatted like this when you have the first 3 columns as genomic coordinates instead of the array identifier and then 2 placeholder columns. So, oh, I have an example right here. Phenomenal. It's like this. This is a bed file. The first 3 rows specify, first 3 columns specify the genomic coordinates, and then the last column right now is specifying the gene. So if we want to look at bedtools, right, it tells you that there are so many different things it can do. Right now, we're going to look at intersect. And if we look at intersect, oh, sorry, you can't pipe to less. I don't know why you can't do that. Bedtools intersect. You could read the documentation, but pictorially, I think it'd make more sense to look at it like that. So if you bedtools intersect, you'll see this picture, and you're going to give bedtools. It needs at least 2 arguments. The first one is -A, which specifies the base file to which you want to find intersections of. And then you can specify multiple -B parameters, which basically are the files that will be used for finding the overlaps. So this one, the blue one, is -A, -B is the orange one, and then the default is that it'll just find the overlaps of A that are the overlaps of B that are within A. You can kind of think of it like that. So you see that these, excuse me, are overlapping here, overlapping here, overlapping here, overlapping here. And then there are other flags that you use for output, like WA or WB, that allow you to control what's actually shown. So if you don't include WA, you'll see you'll basically just get both of the regions that are overlapped. You only get the regions of A that are overlapped with B. But if you do WA, you'll get the entire region of A if it has any overlap with B. So when you have WA, it prints out this entire region because there is at least one overlap of B that overlaps with it. The same thing can be said with this right here. And then this might take a while, but I'll show you what it looks like. Basically, what I'm overlapping is I'm overlapping this reference. That was bad. And this reference is just all of the CPG locations in HD19. And so if I overlap, and remember these regions stored in the hepatocyte.bed are, they're not CPG locations because they're longer than 2 base pairs, but they correspond to gene locations. So you'll see that although we only had 1 entry for chromosome 11, expands to about 5. And then the same thing goes for chromosome 12, 13, 19, and the other, and then 5 as well. And then you can control the output, right? So if you want to also output, if you want to output A, so long as there is an overlap with B, you want to output the regions of A, the entire regions of A, so long as there's any output with B, you'd specify WA. And if you want to output the overlapping regions of B, you'll print out you'll include the WB at the end. And I don't think there's a good image of what includes with WB, but it includes exactly what you think it would, right? So if A includes genomic regions, and then B, I'm sorry, A, we're actually giving A to be the CPG locations and B to be the overlapping regions, right? So WA is actually going to be the CPG locations here, the first 3 columns, and then the next 3 columns are going to be those original overlapping gene regions from the hepatocyte.bed file right here. So you can see that these have length of 2, which indicates that they're CPG locations corresponding to cpg.bed.gz right here. And the next 3 columns have longer than 2 locations, and they're constant for a given gene because they correspond to a gene region, which is what's stored in what's given to the B flag, which is the hepatocyte.bed. And that's one basic way of using bedtools intersect. And on their website, they do have examples. They have use case examples at the end of it, right here. Very simple examples of here's A, here's B. We want to find the overlap of A and B, right? If this one's 15 and 20, it's going to find this one right here, chromosome A, and it'll print out what's stored in B, apparently. Okay, does that make sense? Any questions? That was just one function of bedtools and then one use of bedtools intersect. As you can tell.
Can I see the CPG index?
Yeah.
Okay.
All it is a 3-column file.
Yes.
That specifies the genomic coordinates of.
I have a quick question about the CPG location. And I think Wending answered me once, but I forgot about it. The CPG is a C and G. So why there's 3 location lost on the location? I mean, 3 space for 2.
What do you mean?
So one cytosine and 1 guanine. So why need 3 space to store 2 dinucleotide?
He's asking about like the CG nomenclature versus this nomenclature.
Why are you asking why you need 3 columns?
Oh no, why you need 3 space? Like the first row, the 10,468 to the next should be 469 and 470. So there's 3 space. But we only need CPG like cytosine and guanine, like 2. So
Give me a second. So there's a thing with. I'm gonna look this up on TrackHub to make sure I'm telling you the right thing. You're asking why is it 168, 169, then 170?
Yes. So
Okay.
I don't know where to start the cytosine.
We can actually look at we can actually do this right here. This will be pretty useful. If you haven't used the TrackHub before, and I think that everyone's seen it at least, basically a way at looking at locations. Yeah, so you see this right here?
Mm-hmm.
The C starts at you see I plugged in 1068.
Yes.
And then it started here's the reason. Okay, here's the reason. I'll explain it. Okay, you see the whiteboard?
Yeah.
Basically, we have a C and we have a G. And it said that, I'm sorry. We have a C and a G,
Yes.
right? But it put the number 10469 here, which indicates that you don't have positions for nucleotides. You actually count between nucleotides, like right here. Yellow will show up right here. These are where you count the positions. So like in computer science, you're gonna base, sorry?
So there's nothing there, but they still count, right?
Yeah, that is the position between nucleotides, yeah. So in computer science, you can have base 0 counting, you can have base 1 counting, and that kind of differs between programming languages. In bioinformatics, we kind of just said screw it to both of those methods, and we do in-between character counting, where this is 10, it's a different color. We do 10, 4, that's a good question, 4, 6, 8, right here. And this is 10, 4, 70. So the reason why we need 3 numbers, well, we don't need 3 numbers. We would need, even if we shifted it, even if we counted on the C and the G, we'd still need 2 numbers, right?
Yeah.
But we can basically, I don't know, man. It's just how we store information about where CPGs are located, or actually it's beyond CPGs. It's how we store information relating to genomic coordinates.
Okay.
Yeah. But when you think about genomic coordinates, don't think too much about where they are, if they're between nucleotides. Just think that I never really think too much about, I always think about relative position when I look at the data. Like for right here, I don't think too much. Okay, I don't think about where this is located. I know that I see the difference here is 2, so it represents 2 nucleotides. It might also be because if you had, oh, here's a reason. Here's a good reason, actually. If you took the difference of this, you'll get 2. But if you put the numbers exactly in line with the nucleotides, then that would actually encompass 3 nucleotides. If you had 10, 4, 6, 8, 10, 4, 6, 9, and 10, 4, 70 under each nucleotide, then when you subtract, it wouldn't fall in line with how many nucleotides are actually being represented. That's the reason. And that's a very important reason. Do you want me to draw something, or do you understand?
You can draw something, so just help me to.
Okay. I just came up with that. So, right, if we had, right, so if we take the difference between this number and this number, we get 2, right? But if the numbers were represented so that it was 10, 4, 6, 9, I'll highlight this in red. And then we had 10, sorry, this one should be 10, 6, 4, 8. Will not allow me to edit it, or? That's annoying. Undo, 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 undo. 6, 8. And if they were directly under the cytosine and guanine, then, and then if we had another, should be this could be a lot easier to move around in here. 10, 4, 70 over here. Then this would encompass 3 nucleotides instead of 2. Because when you do 10, 4, 6, 8 to 10, 4, 70, you subtract 2, right? and then you know there's 2 nucleotides, and then it also encompasses 2 nucleotides. But if it was in line with the CPG locations, then when you do subtraction, you'd actually be representing that with 3 nucleotides. So this number minus this number is 2, but you'd have 3 nucleotides represented here.
Okay. So basically the last nucleotide just doesn't show up, right? So
Does anyone else see this, or? Oh no, did I confuse everybody? Oh no.
No, that makes sense. You're just
Okay.
talking about the start and stop for a CPG, it'd be 2 because we're talking about 3 nucleotides, right?
2 nucleotides.
It'd be 2 because, or it would be 3 because you count between.
Okay.
We count 2 nucleotides, right? It's a C, phosphorus, G, right?
Yeah.
But we do that, we count in between nucleotides so that when you take the difference of a region, you get the number of nucleotides, right? So if we had it represented like this, where you had the index exactly below the C, exactly below the G, exactly below the N, then when you take the difference of this, you'll get 2, but you're representing it with 3 nucleotides here. And that's the problem.
Okay.
Tony, do you see this?
Yeah, I think that makes sense.
Okay.
So
Yeah,
you're using 2 that way. When you take the difference, it shows up as 2 nucleotides instead of 3.
yeah.
Yeah, I
Yang,
get that.
okay, Yang?
yeah, it makes sense.
Okay, that's a good question because it's a good question because it took me a while to think about it, and I didn't have a good answer. And I knew that bioinformatics has this as a convention, but it's hard to determine why. And that's a good question. But when you look at these files, I don't really think about like where it's pointing to or what it's representing. I just know that this comes before this. This is the CPG directly before this one. And then there's some space, and then it's this one, and then some space, and then it's this one. So, but a good question. Good question. Okay, well, we're a little over an hour now. I will put the updates I made to this file on my GitHub, and then I'll send out the recording and the channel and all that fun stuff. But yeah, thank you. I appreciate all the questions, everyone. It helps me to kind of figure out in what direction I can take this, and I'll get some feedback in a bit and decide about next week or whatever. Thanks, Michael. And we can talk about more topics. We kind of, we're kind of whipping AWK to a pulp, so we can do more of tbmate, or we could do bedtools as well. All right. Well, thank you very much. I hope you all have a good night. I'm done for the day.
Thank you. That was very helpful.
Thank you so much.
Of course.
Thank you.
All right. Take care, everybody.
Have a good night.
Good night.